Thank you very much, Dr. Skandamis. I would like to thank the organising committee for allowing us to present our work. And of course, I would like to say that my presentation is not going to be equally interesting as the two previous presentations by Mr. Zampelas and Mr. Thomaidis, because it's going to be more technical. Can we go back? So as you heard from Mr. Skandamis, I work in the Biomedical Research Foundation of the Academy of Athens, and I'm dealing with proteomics. So, proteomics research unit. But for today's presentation, I'm going to start in a different way. We're going to speak about personalized medicine. What is personalized medicine? And where does that take us? Where does it lead? So far, when we spoke about personalized medicine, we referred to a medical approach in which each person was treated in a unique way in relation to the treatment suggested. So it's not a homogeneous approach, but we look into the various characteristics of each person, and then we follow a specialized, a personalized approach that is suitable for this person. So this is the vision that we have for personalized medicine. The vision is to get from the population to the individual, looking into various parameters. So we do not have mass treatment, but we have personalized treatments that have to do with each individual on a specific level. In the past, we had, the old way. We had the patient's tissue sample, we had the pathology, and then we identified the treatment. The new way is using the technology that we have available, such as proteomics and genomics, that produce mass results. For the proteomics in relation to the proteins of an individual, genomics for the various DNA aspects, and the genes, we can classify patients, and given that we have that many results, we can individualize afterwards the results that we get per patient. So for this personalized medicine, we have the genome, the transcriptome, and the proteome, and then we have the epigenome, the metabolome, and the microbiome. A combination of all of these gives us an idea about an individual, and then based on that, we decide on the treatment. This is the clinical decision data that we have for personalized treatment. Again, this is very complex, but keep it in mind that it combines lots of data, even ethnological data and the various characteristics of each patient. So we reach what we call the decision tree. We combine all of that, and then we get to the result, and we opt for the treatment. This is all fine. All this concept, we've tried to carry that over to the food industry, thinking of food as a patient. So we got the entire concept of all this personalized treatment, and we used that on foods. So thinking of a piece of feta cheese as a patient, and then for that we want to combine as much information as we can in order to have more and more characteristics that then we can evaluate and monitor. This we called trophometry, so the metrics of food. The term trophometry, which of course you can understand being Greek because it comes from trophy, meaning food, and metro, which is analysis, refers to all mass spectrometry methods that can be applied to the analysis of a food. This is things that we do in the laboratory. And this is all applied to the analysis of a food. So we get all the results and we combine that into a sample of a food in order to conclude on its content. So based on the trophometry, we get the trophometric fingerprint from all this obtained information. What is that? Basically, we have the various categories and types of spectrometry, such as mass spectrometry, multi-TOF, or the high-resolution MS, or nano-LC, and all these types of technology combined together give us a list of proteins, and then through some processing, we can get the data that we need. What we get each time for each sample of food is the trophometric fingerprint. This is a unique set of pieces of information, unique to this food, apart from the proteins and the peptides that we get from the spectrometry. This can be enriched with a lot of other types of information, such as the metabolomic, the texture, the flavor, the smell, and so on and so forth. And we can analyze all of that. So briefly, this approach is a typical approach. We have the sample food, then we have the extraction of the molecules we're interested in analyzing, the identification of proteins, peptides, metabolites, and lipids, etc. And then afterwards, through the spectrometry method, we get the result. Some examples and applications so you can understand what it is we actually do. As mentioned by Mr. Thomaidis and Zampelas, of course we have looked into feta cheese because it's a very important product for Greece. We try to answer three basic questions based on trophometry. And the three questions are as follows. The first one, is it a white cheese feta? Because you get a piece of white cheese, you get that in the laboratory, and you ask yourself, is that feta cheese or not? Then, number two, if a feta sample is adulterated or not. And then the third question is if a feta sample is protected designation of origin or not. There are numerous other questions that you could ask yourself, but we started with these three, trying to see whether we can answer them based on our method, and then we will see. So we analyzed various types of feta based on the spectrometry from various areas of Greece, and we know that they produce protected designation of origin. A huge number of samples. No need to go into that. So the first step, what we got is we designed the Feta reference trophometric fingerprint database. So we got all the information we needed, and we have this database that we set up with all the proteins and of course the peptides as well, the peptides from various samples of feta from all over the country, from all over Greece. So the various regions, Cyprus, Macedonia, Thrace, Thessaly, Steria Ελλάδα, Peloponnese and Lesbos, you can see that there are no big differentiations in terms of the proteins on a regional level. 519, 517, so the differences there are not that significant. And then we created the same type of data, and then we created this type of map. A map of the database that we have for the feta cheese. It looks like a flower. We could call it a feta flower. And you can see that in all samples that we got from the feta cheese, there are 489 common proteins. So if you have a sample and you want to characterize that as a feta cheese, it needs to have at least these 489 common proteins, because these are in common. This is the proteins that we identified in all the samples that we analyzed, and it's a lot, you understand, it's a lot. So if you have a sample of white cheese and you're trying to understand whether this is feta cheese or not, then this sample should contain at least 489 common proteins. And then you realize that you have the various areas around, okay, Thessaly, Epirus, etc. And there are various different types of proteins. So apart from these 489, there are an additional 35 or 28 or whatever. And you can see 26 proteins in all samples from Thessaly, 16 in all samples from Steria Lava, and so on and so forth. So you have this nucleus of proteins, 489, as I mentioned, and at the border you can see another set of proteins that allow us, again, to determine the origin of a sample of Feta and understand which region of Greece it comes from. So this result is a first result that we get from the application of trophometry. Combining that with other types of data, we managed to have the so-called trophometric fingerprint for the Feta cheese. As you can see, it contains a big number of analyzed molecules. We have the proteins, peptides, many metabolites, and some lipids as well that appear in every sample of Feta, different, as you can see again, from one another. So all this data, all that we've managed to do here is what gives us the trophometric fingerprint, the reference, I would say, trophometric fingerprint for the Greek Feta cheese. Question number 2 that we asked ourselves, the Feta adulteration, is there some kind of fraud here or not? And again, to supplement on what was mentioned by Dr. Thomaidis and Dr. Zampelas, we try to see what different things we can find in the types of milk used to produce the Feta cheese, because you know that Feta cheese is a product characterized with specific guidelines as to how it's produced. It should not contain milk from cattle, but it's mainly this type of milk with 30% goat milk. So from sheep and 30% goat milk. So here you can see the spectrum, and there's a peptide here that shows us that the milk that has been used derives from small animals, and you can understand which animals gave you that milk. Then we have other peptides 2 and 3 for milk that we get from sheep and milk that we get from goats. And then there's another peptide. As he moves away from the microphone, we cannot interpret. The speaker needs to speak close to the microphone. So the various ruminants that we have, and a peptide which is characteristic of cow milk, etc. So you understand, based on these peptides, the origin of the milk. Apart from the qualitative analysis, whether it contains goat milk or not, you can have a quantitative analysis in relation to the peptides appearing in the spectrometry. So we try to answer the basic questions. You can see the answers, and then we move on to the second example that we have for the identification of the origin of different marine organisms. So what we tried to do in this case was to identify the origin of this shrimp from various areas of Greece and sardine. Again, because shrimps and sardines from some areas like in Kalonine, Lesbos, or in the Gulf of Ambrakikos, these two products are very important to the economy. And then the third example for the trophometry has to do with the identification of the subspecies and the origin. We have two very similar products here, types of fish, the Mullus somnoletis and the Mullus babatus. So it was a challenge for us trying to understand two subspecies based on the metabolites and the peptides. We try to distinguish between the two and see whether it came from one species or the other. So we have the database, and then when we get blind samples afterwards, we ask ourselves the same question and try to answer whether this is Mullus somnoletis or Mullus babatus. And another application that we have of trophometry, a very recent one, has to do with the identification of the pest of small ruminants by the mass spectrometry in ovine milk. Because the small ruminants, you can detect that in PCR testing, etc. So we've identified a set of proteins from small ruminants in milk, and then that leads us directly to the identification of the disease. As you know, for experts, this type of disease has to be declared in the Ministry of Agriculture and Development, but there's a very fast method of identifying that, this pest of small ruminants. Within 2.5 to 3 hours, you can get the final results. Before I finish, I would want to share with you the overall concept of the last few months, the One Health. Where does that all lead us? It leads us to a common treatment of health, whether we're dealing with animals or with humans, or whether it has to do with COVID or the pest of small ruminants. Because again, COVID, as you know, started from a disease that was traced in animals. And this is a very recent approach. There will be an article either to be published very soon, or maybe it was published last month, on the use of proteomics in the approach of One Health. Common health, One Health, unified health. There are various terms used for that in Greek. We have not decided which one we're going to use. So we have the various infections, the zoonotic infections, but all that comes under the same framework. It's the same mindset whether we're dealing with animals or with humans, because pharmaceutical products might in some cases be used both in animals and in humans. That's the end of my presentation. Thank you very much for your time. 2 final remarks. That all applications need to be integrated, and the term trophometry, which can answer crucial questions on the study of various hypotheses in relation to food. Thank you. Thank you all for listening to me. Thank you to the organising committee.